Rajat. Never mind.
Okay, so I just started the recording of the class. Basically, as soon as the class is over, my computer will automatically kick out a recording of the Zoom meeting, and I will pass it up to Eloy, and it should be up on the YouTube, I think, before the end of the day, probably.
Okay, sounds good. So I can just sign off and I can just return to the class tomorrow then?
Yeah.
Okay, sounds good. Thank you.
Of course. All right, so everyone out there will notice that in chat, I posted a link to the Google Drive. That's going to be the slides I'm using today. All right, let me just get my PowerPoint queued up.
No, I can't see your PowerPoint.
What?
No, you can't see your PowerPoint. I can't see your PowerPoint presentation.
Oh, yeah, it'll be just a second till you can see it.
Yeah, okay, cool.
All right, now people should be seeing my PowerPoint.
Yes, we were able to see it.
Okay, perfect. All right, so I think I can get this thing started then. All right, so first I want to welcome everybody to the new Workshop 11. Couple announcements and other sort of formalities to get out of the way before we get started, and also give people who are running a little bit late or having trouble connecting a chance to do so. This is a new workshop, so there's probably going to be a few parts of it that may not be completely smooth. But when I was going over the previous workshop, and the reason why the workshop has been on hiatus for a little while is the previous workshop, while it was good, a lot of the technology in it, as I was going over it, is extremely outdated. So basically what you're going to be seeing is just about a completely new Workshop 11. It's been so a couple of things that I've done versus the old format, if anybody here saw that or was wondering. The new workshop has a much bigger focus on understanding the process end to end and understanding the fundamentals. The previous workshop did a lot of analysis. It basically had you doing a lot of analysis, although you wouldn't necessarily go into depth about how to understand them. With microbiome, I've noticed that, so microbiome is also what I work with in my day job a lot. And one of the things I've noticed is that understanding of microbiome techniques is a little bit lacking, even in a lot of people who are running it. So I'm actually aiming for kind of a deep level of understanding of the fundamentals versus necessarily being able to do a bunch of fancy stuff. I will talk about things like alpha and beta diversity, and probably principal coordinates analysis and that kind of thing. But for the extremely fancy plots, Those change every day, and usually online you can find a manual for how to do them. So the data sets for this class are new. Again, an advantage of the fact that I work with this for my day job. I have access to some data sets, including some pretty standardized data sets that I'm pretty free to use. And I include two data sets here. One of them is using actually the Bioinformatics standardized mock microbial community. So that's a relatively low complexity. It's 8 microbes, or I should say it's 8 bacteria and 2 yeast. Since we're doing 16S, primarily we're going to be seeing bacteria. And that gives us a low complexity sample with completely known inputs. We also have a sequencing run from stool. So stool is a more complex community. You don't actually necessarily know everything that's going to be in it. And a fair warning, I'm sort of making this up as I go along, although not completely. So we're scheduled for 3 hours per day. Nobody wants to do 3 hours per day of Zoom meetings. So realistically, I'm aiming to get us out between 2 and 2.5 hours. The slides that you get before each class are going to be draft version. And the reason for that is the only way I can really work out the timing of the class and figure out where people ask questions and that sort of thing is I just have to run it and see basically what my stopping points are. So you got the day one slides in the link. Chances are that I'm probably not going to get through all those day one slides. I have more than I need. And that's perfectly fine because the ones I don't get through will wind up on day 2. And as I say in here, if nothing else, I remove all the content teaching about 454 sequencing. So use the raise hands button for a quick show of hands. Who in this class is familiar with 454 sequencing? Okay, I got one hand. Yeah, 454 sequencing is basically like the Betamax of sequencing. And if you haven't heard of Betamax, that even gets the point across more. If you have heard of Betamax, you already kind of get the point. 454 sequencing machines for that went off the market years ago. And I think last year the reagents themselves went off the market. So basically nobody's going to be doing 454 sequencing anymore. It was kind of obsolete several years ago. Its importance to the microbiome community is that a lot of the early work on 16S was done using 454. So you'll find a lot of early work with 454 sequence. All right, now on to the show. So the way I tend to structure my classes is on the first day, I have it very light for the computational portion. That's for a reason. And I'll get to it in a moment. But basically, I want to make sure people are able to access their computing resources. All right, so introduction to myself. In terms of my relevant training experience for this class, I started off as a bench scientist. So I actually started off doing basically no bioinformatics. I did half my undergrad, my PhD, and my postdoc at UCLA. About halfway through my postdoc, I got heavily into bioinformatics, which is what I'm doing here now. And I've worked in a fair number of fields, including things like microbial pathology, immunology, a lot of genetics and molecular biology and metabolism, and a little bit of cancer biology and that kind of thing. So I have to go into affiliations, especially for this course, because there is just a little bit of a conflict of interest that I want to state right at the front. Right at the front. So my affiliations are both UCLA and Zymo Research. And I say here I have an appointment of some kind in MCDB because that title is actually in the process of changing right now. So I think I'm still technically a 0% project scientist, but due to the increasing amount of teaching responsibility I've been getting, MCDB I think is going to be making me an adjunct faculty member soon, although I don't know exactly when. I'm also a scientist at Zymo Research on the bioinformatics and microbiome teams. So this actually makes a little bit of a conflict of interest for me because Zymo Research sells microbiome products. So we sell microbiome prep kits, we sell microbiome library prep kits. And because of this, you will actually see me actively avoid discussing specific brands of stuff that we sell. with the one exception is I will be referring to the Bioinformatics mock microbial community, and that's because one of our samples comes from that. I mentioned before that we have a low complexity sample, and that's basically what we use for a low complexity sample, so I have some reads from that, and I'll be mentioning that product as the source. The other thing I'll mention is Zymo also, and actually this is stuff that typically I'm the one who did it anyway, I'll be discussing some free open source software provided by Zymo Research. The nice thing is it's free and open source, so this isn't a big conflict of interest because nobody makes any money off of it. All right, so a couple of assumptions I'm going to make about you. I'm going to assume that you have some experience working on the command line. I'm going to assume that you are able to log into the Hoffman 2 computing cluster, and I specifically mean you know how to do this already. Your account is in good standing and active. So who out there has not yet logged into the Hoffman cluster? Just do a quick show of hands. All right, and I'm going to actually, let me see if I can find that. Sorry, just give me 1 second. My Zoom chat disappeared on me and I'm trying to get it back. There we go. All right, so people who said that you have not been able, that you have not yet logged into the Hoffman cluster, please do so. Also, is there anybody out there who, I'm gonna assume you have some understanding of sequencing as well. So is there anybody out there who has basically no experience on the Linux command line? Okay, so that might be a problem. Make sure that you ask questions. If you haven't worked on the command line before, so I make this class with the assumption that you've at least gone through workshop 1. Is there anybody out there who has no familiarity already with next generation sequencing? Okay, is there anybody out there who has not yet worked with sequencing data? Okay, good. I'm seeing no hands being raised. So, okay. So if you're not familiar with the command line, that's going to take a little bit of getting used to. I have a little bit of review of that, but make sure you ask questions because that's an easy way to get behind in this class. All right, so this is the part where I'm going to have everybody introduce themselves. All right, so just a couple quick questions to answer about yourself. And I think people can unmute themselves here. If you can't, just somebody say something in chat and we'll figure out what else to do. Or actually, I think the easiest way to do this probably so it doesn't become a free-for-all is I have some questions up here. So just put your, send out your answer in chat to everyone. Because I find that typically after these courses, my students tend to still work together on things since people are usually at about the same stage. So it's usually a good opportunity to meet other people around UCLA who are doing the same thing. So the questions are just, what's your name? If you are a UCLA affiliate, what stage are you in? So undergrad, grad, postdoc, etc. What department and lab are you in? And what are you studying or planning to study? If you are from outside UCLA, then what's your affiliation, your role, and what are you planning to study or work on? And I'll give everyone a few minutes to just get that into the group chat. think I have everybody, or else they should be getting in. So, all right, and hello to Timothy Moore, because I think I saw you in the lab meeting I presented last week. And So Yoon Kim, which lab are you in at Cedars? All right, Sarah, welcome to the class. And oh wow, you're working on DGR evolution. Okay, I've been at UCLA forever, so I actually knew Sergey while he was here.
Oh, nice.
What? No. All right, welcome to Vivian. Welcome to Eva. And very cool that you're doing built spaces. I know a few people who are working on the microbes of built spaces. I also like the fact that I'm seeing kind of a variety here. So it used to be that in a microbiome, you pretty much see like 90% of the people were working on human, and almost every one of them was working on human stool samples, so gut microbiome. I'm glad to see that's changed around. One of the things I'm going to talk about later is the fact that our databases are just horribly skewed right now, where we have tons of coverage of a few groups of microbes, and we have horrible coverage of a lot of environmental microbes. All right, welcome to Karen Zhang. And welcome to Farzaneh. And hello to Wayne. So Wayne I already know from some water meetings. And hello to Lewis and Jonathan Lynch. Oh, from the Shao Lab. Shao Lab does really cool stuff. You can actually see Dr. Shao in some TED Talks and those kind of things, talking about the gut-brain axis. So I don't talk a lot about that in this course, but there's a ton of research right now on how the gut microbiome affects the brain and mood and all that other cool stuff. And Eloy, yes, it's cool if you take attendance after this. Okay, and yeah, Jing, also welcome. I haven't met you, but I actually know Tantano's lab really well. I did my PhD in Steve Young's lab, and we were on a grant with you guys. So, oh, we have somebody else rotating in Elaine Chow's lab. Awesome. Yeah, that lab does cool stuff. All right, and Georgia, were you in one of my previous classes? I don't see your face, obviously, but your name looks familiar. Okay, yeah, that makes sense. All right, so I think Eloy is about to take attendance right now, so I'll give him a minute to do that, and then I will pick up with the course.
All right, hi everybody. So I'm gonna leave a link in the chat room. So that way you could take attendance. Also, I also need to see a show of hands for those who have completed this form. Okay. So can you go ahead and do that right now? Blah 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 blah. So I'm mumbling too much. Okay. Participants. Oh, my bad. It's sent it to Weinstein. All right, let's try that again. All right, here's the link. So after you complete the form, just raise your hand on the participant window, and that will show me if you have completed it. So I lowered this down a little bit. There we go. All right, cool. Let's give it another minute or two.
Mm-hmm.
Yeah. Cool. Cool. And then I got two more. Okay, okay, thank you. Barzaneh. That's the last one. All right. Yeah, this is technically also goes towards anybody who's taking this class for 275 A or B. This goes to your credit. So please take this seriously. All right. All right, let's look at most everybody. All right, cool. Got everybody. All right, thank you. Michael, you can lower their hands now.
All right, there we go. All right, I think I can pick up the course again. All right, so again, as I mentioned, day one tends to be fairly heavy on going over concepts, and it tends to be fairly late on computation. In fact, I think the only thing we're really going to be doing today is we're going to be setting up a Cyberduck connection to the Hoffman cluster. All right, so one of the first things I want to get into is metagenome versus microbiome. And what does that mean? And a lot of times you'll hear me and a few other people use that term interchangeably. And I think generally it can be used interchangeably. There's certain cases where kind of in strict sense, you know, people will say like, I'm referring to something in a strict sense. So generally those terms are going to be used interchangeably. But I think what people are most familiar with in terms of microbiome or metagenome is looking at basically the microorganisms of the human gut. especially when people talk about a metagenome. Metagenome is really also looking at just the entire genomic content of some place or some, or some mass or whatever. So in theory, the human gut can also harbor, it generally shouldn't, but it can harbor multicellular organisms, so people can get things like tapeworm infections. And I know that's kind of gross, but just go with me here. So, and what's also interesting is a lot of worms, almost certainly including tapeworms, are going to have their own microbiome. These are microbes that are associated specifically with a worm. And all of these things are going to have DNA. So the bacteria are going to have DNA. There's also some yeast cells in there, or some fungal cells. Those are going to have DNA. The worm's definitely going to have DNA. And the worm's microbes are going to have DNA. So if you start talking strictly about microbiome in the strictest sense, you should just be looking at the human gut microbiome and probably the worm microbiome too, since that's now microbiome in there. If you start talking about full metagenome, sometimes people will be just meaning the microbes plus maybe the worm, because that's not host. Some people are actually going to even include host. And the simple fact of the matter is that if you're doing full shotgun metagenomics, you pretty much have to work to exclude the host. So the host tends to be a big contributor, so you will pick up host signal. And then sometimes also, maybe you're going to be even talking about the virome. Virome is something a lot of people don't like to get into, but we know it's there. So I know somebody, I think it might have been Yajing, mentioned having worked in mucosal immunity. And something that a lot of people don't always appreciate in mucosal immunity is it's pretty well known that mucus contains, mucous membranes contains a lot of IgA, especially in the gut. There's a lot of IgA on that surface. And that's just antibodies against stuff that you're going to be exposed to on the surface. There's a whole other group there, though, that isn't always fully appreciated, which is mucus tends to hold a lot of bacteriophage. And those bacteriophage basically go in and kill bacteria. So maintaining bacteriophage is almost certainly part of mucosal immunity. And so the virome is actually going to play with the microbes. And sometimes, for the sake of full metagenome, people will specifically say that you can't be including host, but you pretty much, so when somebody says full metagenome, it sometimes pays to ask specifically what they're including and excluding from their definition. All right, so one of the basic principles to understand, as I kind of got into, is everything is somewhere. And I know I have at least one person in the class who's studying epidemiology. Is that right? I'm going to go back through, but I think I have somebody in this class who's studying epidemiology. Karen Zhang. So specifically at Karen, although if anybody else happens to know this as well, does anybody know the significance of this triangle I drew on the globe? Anybody? Has anybody ever heard of something called tsutsugamushi or scrub typhus? Okay, there's some people who've heard of scrub typhus. Have you heard of it because you were either exposed or from a place that was where it was endemic? All right, anyway, scrub typhus, it kind of sucks. Fortunately, thanks to modern antibiotics, it's not as big a problem because basically a course of doxycycline makes it go right away, but you get really sick with it. So scrub typhus is spread by these mite larvae, and basically it goes by a couple names. So the English name for it is scrub typhus. The Japanese name for it, which it also frequently goes by, is tsutsugamushi. Tsutsugamushi literally means bug illness. And so if you're in this triangle, especially if you're going around scrubland where there's, a lot of grass and marsh, you're going to come across these larval mites. They also go by the common name of chiggers. And they're really unpleasant. They itch a lot, but also they have a microbiome. So if you just go and look at this thing, you're going to find there's microbes that live there. I have sort of the nice, cool microbes that are there and harmless and blue. And I have this sort of oval-looking one, this kind of gram-negative oval-looking one in red, because that's the one we're really interested in, and that's specifically the one that causes tsutsugamushi. In fact, that's actually the name of the microbe. Oh, yeah, that makes sense. I should have guessed from your name. So you were born in Korea, and yeah, Korea is like right around the middle of this triangle. Yeah, so we have this one here. What's interesting is that in these chiggers, and actually throughout the stage of this mite, this is part of the normal microbiome. This is something that just lives with it, and it's actually completely cool with it. So these insects have a life cycle. Obviously, they go, like most insects, they go from egg, they go to larva, they go to nymph, they go to adult. And this intracellular bacteria basically lives with it at every stage. I was trying to look to see if it gives some advantage, because a lot of times you'll find these bacteria that basically just live with it at every stage will typically give the organism some advantage. I couldn't find one, although I would be surprised if one day we figure out there is one. But these things are, if they show up in mites, they're basically a normal part of the microbiome. And they don't really seem to cause any trouble. They just pass through with every generation. Occasionally those larvae bite a rodent, or they bite a human, at which point this becomes part of the rodent's microbiome. Now, if this becomes part of the rodent's microbiome, that's a problem. It turns out that Orientia tsutsugamushi, the causative organism that I'll just call tsutsugamushi, it doesn't want to be part of this microbiome, and the rodent doesn't want it as part of its microbiome. So this winds up causing an illness. This winds up causing scrub typhus in the rodent, or if it's an unfortunate human that gets bit, then it's scrub typhus in a human. This is occasionally also how tsutsugamushi can actually jump and move around and that kind of thing. But so, for example, in sort of a family of mites that aren't infected, this is how during a feeding it can jump into a new family, and it'll just go into the cycle through there. So it spreads pretty widely. The rodent or the human is an accidental host. In the case of the rodent can either survive the illness or the rodent can die from the illness. But either way, it's kind of a dead end for the microbe. So the microbe doesn't want to be part of this microbiome. The microbe wants to be part of this microbiome. The rodent doesn't want it, and when it shows up, it causes all kinds of havoc. But it's kind of important to think about, because for many years, the thought of microbes were just associating them strictly with, filth and disease. But realistically, everybody in this photo has a microbiome, and most of the time it's just doing its thing and it's not being harmful. And in fact, the same organism in one place can be just completely normal and harmless, and another place can cause all hell to break loose. All right, so course overview. Again, this is rough because I'm still working out the timing of this course, and I'm still trying to smooth it out. So sections might get rearranged, stuff may get moved between days, that sort of thing. So day one, I'm going to be doing a bioinformatics review for people, because I know some people either don't have a lot of familiarity with the command line or haven't been at the command line for a while. Talk a little bit about the course goals. I'm going to talk about types of analysis, and I think this is actually the most important portion right now, because again, I work at Zymo, so I also deal with, along with design, my main role is making making applications to help make microbiome analysis better, or make analysis to help make our microbiome products better. But I also deal with tech support, especially for microbiome issues. So I kind of know the issues people are running into. And one of the big things that I've seen is A lot of times people will wonder why they're not getting the answer they want for their microbiome, and it turns out they're not asking the right questions. So I'm going to talk a lot about types of analysis. I won't be getting to starting materials today, but I will be getting into potentially a few other things. All right, so the first topic I want to do is a bioinformatics review. And the question comes up, what is bioinformatics? Because it has different definitions for different people. So I like to say bioinformatics is kind of in this region right here. So if you draw a Venn diagram with biology, computer science, and mathematics and statistics, you'll see bioinformatics kind of lives right in this middle. Some people are heavily skewed in one way or another. So you'll find, so if anybody is purely in one of these circles, they're definitely not doing bioinformatics. If somebody is doing biology and math and stats, a lot of times people just refer to that as biomath. So that kind of has its own field over here. But especially in this intersection here, you'll find a lot of people who are doing biology and computer science will consider it bioinformatics. And people who are combining all three are doing bioinformatics sort of in the strictest sense. And in fact, the better you are at all these three, typically the better you do with bioinformatics. So bioinformatics review. And the first thing I want to go over is sequencers, because this is a lot of times the first thing people start wondering about when they're going to do an experiment. And there's now a whole variety of them. In fact, those who took my workshop 2, this is probably going to become a new slide in my workshop 2, because some technologies have basically gone away. So I talked about 454 earlier, and I'll talk about 454 again. You don't see 454 on here because 454 doesn't exist. It's pretty much been 100% replaced by Illumina, and it's kind of gone the way of the dodo. So just talking a little bit about machines here. There's the nanopore, and who out there has seen one of the min ions in person? All right, I got one hand. So these are not drawn to scale. These are definitely not drawn to scale. In fact, if I drew these to scale, you wouldn't be able to see this thing. So for reference, this plug back here is a USB plug. Basically, the min ion is great. You can put it in your pocket and take it with you. And this is really useful actually right now because so min ion has some advantages and disadvantages, and I'll go into those in a minute. There's the MySeq. In this machine, I single out because these three in the middle are Illumina machines, but these are Illumina machines you're likely to come across, and the MySeq has some very specific properties that give it advantages for microbiome analysis. And at the end, we have PacBio. So Illumina-Seq is all the same general sequencing technology. Most Illumina machines will give you up to 150 bases, but you can do it paired in, so you can get up to 300 bases of read, 150 in each direction. The exception to that is the MySeq, which can actually do 300 base reads in both directions. And that becomes super important for microbiome because if you get a 600 base read instead of a 300 base read, that actually enables you to do certain things that you wouldn't be able to do otherwise with 16S sequence. So nanopore, it's super cheap to get started with this. You can get one of these things for about $1,000, and that'll give you access to sequencing. It's more expensive per base of sequencing, so kind of annoying, but again, you have something that fits in your pocket. This is also great. So ironically, sorry, I just have a message coming in real quick. Okay. So ironically, I was actually going to be going to the Democratic Republic of Congo. I was going to be running bioinformatics workshops out there. And this was going to be for people who are monitoring disease outbreaks in the jungle. So things like Ebola and, things like Ebola and Nipah and other nasty microbes that go bump in the night. Except thanks to an outbreak that seems to have originated in China, that trip got canceled. But we love these things. And in fact, in the current outbreak, the reason why people were able to get the entire viral genome and are able to get so many different variant viral genomes done so quickly is this little guy right here, or some of its bigger versions. They're probably using the bigger versions because they're taking it to labs. But I could basically walk out in the jungle with this thing in my pocket, and as long as I have access to a decent amount of power and a few basic lab supplies, I can sequence basically in the bush. So it's more expensive per base. It can take up to 2 days per run if you want to go full length. A lot of people don't go full length because you kind of get diminishing returns. Read lengths for this thing, the longest read I've heard of is about 2 million bases, but a typical read on this thing will be 5 to 7, be about a 5 to 7 figure number. Makes a huge amount of errors though. I believe this thing is, error estimates on this thing I've heard estimated between 5 and 15% of bases are missed calls. So you can assemble a genome off that, but you definitely won't have a polished genome. There's the MiSeq. This one is more expensive. I think a MiSeq is going for about $130,000 now. Cost per base, it's definitely more expensive than these other two machines per base. You get the longest possible reads, and it makes relatively low numbers of errors. So this one's error rate somewhere closer to about 2%. Although as you get further along in the long reads, and I'll talk about this later, you start to accumulate more errors. The HiSeq, this is still a popular machine. It's still in a ton of cores around UCLA, so I'm going to talk about it. Illumina doesn't really make this one anymore, but you're still going to come into contact with them. So as far as the cost of this thing, this machine costs, you get a nice house in Orange County for this price, for the cost of this machine. Cost per base is super cheap. Cost per run isn't cheap, but that's because you're getting a lot of bases. All these machines will run for up to about 2.5 days. You get shorter reads off of both these machines. So basically the NovaSeq platform is a replacement for this. The NovaSeqs are really big machines. And then finally, PacBio. PacBio combines the long read capabilities, not quite as long as Nanopore, but still very long reads. So this one is still about 2 orders of magnitude above what you get with Illumina. It runs pretty fast, so you can actually do a pretty long sequencing run in a workday. The error rate is incredibly low, although I kind of had that with a little bit of a caveat. In the case of PacBio, you can either do shorter reads and read them multiple times, which will give you an ultra low error rate, or you can do longer reads and accept a higher error rate. So this one you can do a little bit of trade-off, but it's actually fairly nice. The only thing is it's expensive per base. The machine, so all these machines, I think this one is on the order of about $650,000 or so to $700,000. These two are typically going to run somewhere closer to like 800,000 or 900,000. So super expensive machines. With I typically don't go into these pricing issues. However, with a lot of microbiome labs, people are collecting in the field, and a lot of people like to go with this one, at least for initial sequencing. This is also great for genome assembly, and I'll get into that later. All right, so for those unfamiliar with a FASTQ file or a QSeq file, a lot of people are probably unfamiliar with QSeq files, which is fine. I wish I was unfamiliar with QSeq files. So these are just two different ways of storing sequences. There was another way that 454 sequencing stored sequences. I'm not going to get into that because it's 454 sequencing. So QSeq, both of these file formats, you get information in the name. With FASTQ, you're going to actually have a sample name. You're going to have the sample name in the name. With QSeq, you may not. But the file name will tell you things like the lane number, the read direction, the tile number, and the file format. Typically with FASTQ, you're going to get your sample name, your sample number. So sample number comes from a sheet you give the sequencer. You get the lane number. The read direction is important. So the read direction is telling you if you're a forward read, which is going to be 1. It's going to be a reverse read if it's 2. And actually with Illumina, sometimes you'll also get a sequencing file or a FASTQ that has indices in it. So index is a barcode read, which you use when you're multiplexing. And that's just how you run multiple samples in the same well so that you don't have to have all your reads go to one sample if you don't need that many reads. So R1 and R2 are forward and reverse reads respectively. You will also get barcode reads, which will start with I for index. So you'll have I1 and I2. And then the last one's going to be file number, which on any modern machine is always going to just be 001. This exists from back in a time where you couldn't sometimes have files that were more than like 4 gigabytes, so old file systems. And when that was the case, you'd have 001, 002, 003, and so on, so that you could have your read just multiple files. QSeq format. And you see here, these lines are all repeating, so everything is on one line. So the first block of values are going to give you machine ID, run number, lane number, and tile. So tile is just a position on the flow cell. You'll get the X position, Y position, index, and direction. So that's just more information about where the read was and what direction it was and that kind of thing. You'll get your sequence, you'll get a quality string, and you'll get whether or not it passed the Illumina filtering. So whether or not the Illumina machine thought that it was a valid sequence. And now's when I talk about QSeq versus FastQ. So QSeq is one format, FastQ is another format. FastQ basically is a format that everybody expects now, and it's a format most programs expect. So FastQ format. This is one read in FastQ, and you'll notice that in FastQ, one read takes up 4 lines. So you have one line that has a bunch of numbers and other stuff on it. That's probably what I call the housekeeping data. So things basically like, you know, where it was read, that sort of thing. You have another line that's A's, T's, G's, and C's. That's going to be your sequence. There's this line that has a plus on it, and that's pretty much a spacer line. A lot of times it'll be a plus. Occasionally you'll have a plus, and then you'll have some other information on it that's just sort of auxiliary information. And then you have this long, string here of weird-looking random characters, and I'll explain what that is in a moment because that winds up being very important. All right, so what is this? Again, as you'll notice, this first line has a lot of, again, what I call the housekeeping data, telling you things like the machine it was run on, the run, the flow cell, the lane. This is also really cool when you think about it, because if I have a single line of FastQ data, I can find out what machine it was run on, which run on the machine it was run on, which will tell me basically when it was run. I can tell you which flow cell it was made on, which lane in the flow cell it came from, and this will tell me where in that lane it was read. There's also a little bit of additional information. So this is kind of the stuff you find at the end of a QSeq line. And that'll tell you things like direction, whether or not it passed Illumina filtering. Control bits we're not going to worry about because I never see anybody really worrying about that. And then the index or sample. So an index will typically be a sequence, that's its barcode. A sample will just be a sample identifier. All right. So what is that quality string I kept talking about? And after I get through quality strings, I'm probably going to call for a quick break because basically I always try and give people a break right around on the hour. So quality strings use ASCII values to encode 2-digit integers using a single character. Why you do this? Basically because you want to somehow encode a decent amount of information, something beyond just a single digit number. But you need to encode it with a single character, because if you look at this, you'll notice that the length of the basis here is always going to be the same as the length of the quality string. And that's because it's a 1-to-1 correlation. This letter here corresponds to this base. This letter here corresponds to this base. This letter here corresponds to this base. This letter here to this base. This letter here to this base. So the question is, what is it saying? And the way we analyze these things is we go from a character to a number using a very common encoding scheme. We go from a number to a base value adjustment. And I'll talk about what that is in a bit, but that's basically just a simple subtraction. We convert that to a Fred score. Now the Fred score is easy to understand, but translating that to confidence involves either a complicated math formula if you want to be exact, or a very easy rule of thumb if you don't need to be super exact at the moment, if you're just kind of eyeballing it. All right, so quick show of hands. Who out there has seen The Martian? If you have, you will totally recognize this scene. Okay, cool. A lot of people have seen that. Okay, this is probably, I think, one of the best movies to watch if you're in the sciences, just because it's got such kind of like a nice attitude about problem solving. So if you remember this scene, he's trying to communicate with NASA, and they're trying to figure out a way to send messages back and forth. And the first thing he realizes is sending messages where the only response he can get is yes or no, and he could just write the messages through a camera is kind of terrible. So he comes up with an idea of using a numbering system to try and communicate letters. And he has to go dig up something called an ASCII table, which looks a lot like this. So this is how, so computers don't do letters, computers do numbers. Computers really don't like handling letters because they just basically count, and actually computers can only count to 2. But if you string together enough ones and zeros, you can make any number you want. So this is number in binary, this number in hexadecimal, which is base 16, which is what computers tend to like working with larger numbers in. This is decimal, this is what humans like. And we see a whole bunch of characters here. So a couple things I'll point out as features on this. This entire block here that I marked in yellow, these characters don't print. So you'll notice things like the shift key is a character number, carriage return, new line, or character numbers. My personal favorite is character number 7, which is bell. And that's literally telling your computer just go beep. So we have to use printing characters, otherwise they won't show up in the file. So a typical quality score is what we call base 33, where the first character that's allowable is character number 33, which is an exclamation point. You'll find occasionally files will use base 64, which is a very old encoding scheme. I think it was popular with the Solexa machines, which is another, I think that's an even deader sequencing platform than 454, if that's possible. But they came up with the idea of using this as their base value for reasons I'll never understand. So occasionally you'll find these in old files. Typically you won't, but if you're looking at old files, keep in mind your quality score encoding may be different. So again, base 33, base 64. And just an example, I'm going to take something with a value of 73. So this is what you would say if you have a quality score of I. If your quality score string shows an I for a base. The way you're going to calculate it is you're going to take the value for that letter. So again, if something gets a quality score of I, it's going to be 73 minus the base value, which is 33, which is Fred 40. All right, so we have Fred 40. What does that mean? You can translate Fred to a confidence score with this formula. And usually when I show people this formula and I tell people it's easy to deal with Fred scores, this is the response I get. So if you want to get an exact confidence from a Fred score, you have to use this formula, and that's not much fun, but the only people I've ever seen doing it this way are dealing with it on a computer. This is how you actually deal with Fred scores. So if you don't want to deal with this formula, you just use the rule of thumb. And the rule of thumb for Fred scores is if you have a Fred of 0, you have 0 confidence, or your error probability is basically 100% or 1 out of 1. This would be a base call of n. Basically, the machine didn't see anything there that it could possibly call. A Fred score of 10, that's going to tell you have a 1 in 10 chance of error, or 90%. And you notice this is going up logarithmically now. Fred score of 20, that's 99% confidence, or 1 in 100 chance of error. 30, 99.9% confidence, so at this point, 1 in 1,000 chance of error. And Fred score 40, this for Illumina is the typical maximum score you'll get for a for base confidence. So the most confident Illumina will say it is in any given base call is 1 in 10,000 chance of error. When you do alignments, they'll typically go up to 60. So your alignments will say 1 in a million chance of the alignment being by error by random chance. All right, and it's now 10:02. Let's everybody be back here by, let's say 10:15. But there's a good chance everybody at least get up from your computer and stretch a little bit. Stretch your legs, go use restroom, get water, do whatever you need to do. And we will pick back up at 10:15. All right, I think we should be able to resume in about 2 minutes. All right, looks like it's 10:15, so I think we can start picking up. All right, and I have a question from the students. So break times are always a good time to get in some questions of how are the base value adjustments determined? So I'll go back a couple slides really quickly. All right, so base value adjustments exist because in this yellow block, we have a bunch of characters that can't print. So if a character can't print, it doesn't end up with a space on here and you can't read it. So obviously we can't use non-printing characters on here. If one of these guys had a quality score that was a beep, this would look extremely weird. If they had, quality scores that were the new line key, it would look extremely weird. So we can't do that. So all these non-printing characters are off limits to us. And that means the earliest score we can use is 33. So because of that, 33 ends up being our base. And the base score is actually pretty easy to use. So let's say that you have a Fred score of 40. You start off with your base, you add 40 to it. So that'll take you out to 73. So that'll be the I. And if you have a quality score string again up here, and you can see that a lot of these are I because they're good quality bases, or they're good quality base calls. You do the numerical value for this I, which is 73, minus your base, which is 33, and they'll give you 40. So for Eva, does that answer your question?
Yes, thank you.
No problem. That's an easy one. All right, so quick review on computing. So again, for this class, I assume you have already accessed the Hoffman cluster and that you have some experience working in a command line environment, especially if that doesn't apply to you, pay attention here. All right, so I call this part Linux in 60 seconds, just kind of understanding how computers work and the architecture behind them. So kind of at the center of everything, you have what's called the kernel. This is basically the central part of your OS that is just controlling where data is going and what data can be accessed and that kind of thing. So the kernel talks to all kinds of different things. Kernel gets input from your keyboard. It reads files. It runs applications. It sends data to and from your network adapter. Outside of that, you have your shell. And your shell is basically a thing that takes in commands from you written in something that you can at least reasonably understand, like bash, and passes them along to the kernel, usually interpreted into some kind of language the kernel can understand. And then the kernel will do whatever it is you're telling it to do, or it'll tell you it can't do it. And then outside of all this, you, on some systems, have a graphical user interface or a GUI, and that's a wrapper. And a lot of times that'll be translating mouse clicks into commands for the kernel or things that get passed through the shell to the kernel. So when you drag and drop a file, your graphical user interface is what's sort of making the file appear. It's what's handling your mouse clicks. And it's what's saying, oh, okay, you drag this file to the trash can. That's actually sending the kernel a message to delete a file. So the way a typical command is going to work on the command line, you're going to have your kind of command prompt, which will usually be some information, including what folder you're in and your username. You'll then have a $sign, and the $sign is actually the command prompt telling you this is where you put in instructions. You'll give it some kind of a verb, something to do or a program to run. Or occasionally you'll tell it to run a program to run another program, like when you say use Python, use the Python language interpreter to run a Python program. You're frequently going to give it some options. So for example, you may pass it -F and tell it a file to work on. And you may pass it -O as some kind of an option or maybe an output file. So you'll be telling it to do the thing this way. So the command line will pass it to the shell. The shell will interpret it and pass it to the kernel as a set of machine language commands. And the kernel is going to immediately say, do I know what this thing is they're asking me to do? Or do I understand the verb? And then if I understand the verb, do I understand the options associated with that verb? So it's going to say, do I know the thing? If it doesn't understand what you're asking it to do, like you're asking it to run a program that doesn't exist, it'll return some kind of an error. If it does understand what you're asking it to do, it's going to check privileges. So it's going to basically say, do you have permission to execute the file you're trying to execute? Do you have permission to read and write all the stuff you're trying to read and write? If you don't, it's going to throw you an error. In this case, it'll throw you a permission error. If you are trying to do something the kernel understands and it says you do have the privileges to do whatever it is you're asking to do, then it's going to do whatever you tell it. This is both a good and a bad thing. The kernel tends not to have a lot of safety built into it, or the command line tends not to have a lot of safety built into it. So if you tell the command line to delete a bunch of files, the command line is just going to delete a bunch of files. You won't have something coming up saying, do you really want to delete these files? Or, hey, this file looks important. Do you want to delete it? They'll be gone. They also typically won't be moved to a recycle bin. They will just disappear. So a couple of basic Unix and Linux commands up here. If you type in pwd, it'll tell you your current directory. If you type cd, it'll change directories. cd ... So ... is shorthand for the folder directly above the one you're currently in. Less is actually a verb on the command line, and less is a program that will just view a text file. cp to copy a file, and the typical pattern you'll see is copy from source to destination. rm is short for remove, and that'll delete a file. rmdir will delete a directory. It will not delete a directory with stuff in it. That's actually a separate command that I'll show you potentially later on. SCP is called secure copy. That's how you copy files between machines. I'm actually not a huge fan of this command because transferring files is often best done through a program called Cyberduck or some similar program that'll just give you a drag and drop interface. If you run this command, yes, you can do it. It requires remembering a lot of remote file systems, which is not fun or easy. You can gzip compress a file, you can gzip unzip, you can gzip decompress a file, or you can run gunzip, which is the equivalent program. Makedir will make a new folder, and then a couple of shorthands. So dot is a shorthand for the current folder, and again, dot is the shorthand for the folder above this one, above the current folder. The tilde is actually a shorthand for your home folder. ls will list all the files in a directory, and wc will do a count of things like lines and characters in a file. So in the Hoffman cluster, software is located all in this one folder called /u/local/apps. If you want to use software that is not installed on the cluster, you can pretty much put the executable in the home directory, or if you want to put a software package on there, which is something we may be doing later, you can store it locally and run it locally from your home folder. You can't write to that /u/local/apps. Only the administrators can do that. If you're trying to install software that requires administrator rights for some reason, you have to basically contact the cluster administrators and ask them for permission. They will generally tell you no, but they're usually pretty nice about it. They'll tell you no, and they'll explain that there's actually some way to run it without requiring extra permissions, or there's some specific reason they don't want it on the cluster. All right, so if you're on a Mac, the way you're going to log in is you're going to use SSH. If you're on a PC, you can SSH from your command prompt now. They've started making Windows have a lot more Linux type functionality because Microsoft realized they're not winning this one. But as an alternative, you can use something like PuTTY, which is very nice. I personally use something called MOBA Xterm because I find it's more feature rich. But for the course, we tend to teach PuTTY, and I can definitely support that. I've used it before. So this is typically what you see when you log in. Couple things to note. If you SSH through a terminal, when you enter your password, you won't see anything happen here. So you'll hit a key, you won't see dots appear, you won't see the password appear. The key is basically just being sent to the remote machine. It's a security feature. The other thing that won't work here is the backspace key. So if you realize that you accidentally hit the wrong key, don't try to backspace to fix it. The best thing to do is hit enter, realize that you have the wrong password, and it's going to give you an error saying wrong password, and then re-enter it correctly the next time with a little bit more care. I'll say this a bunch of times, don't compute on login nodes. If you can, so there's 4 login nodes, and these handle connections between somebody SSHing in from the outside world and the cluster. And if you want to do computing on there, that's fine. You just forward your login session to a compute node, and you get some resources of your own on the compute node. If you start trying to do compute heavy operations on a login node, the login node suddenly finds that its CPU is really busy, and everybody who's connected to that login node suddenly starts to lag, and they get really mad at you. And the last thing you'll get once your login is complete is you'll get a command prompt, usually with your username, the computer you're connected to, and your folder, which in this case is a tilde because I'm in my home folder. And a lot of times when you first log in, you'll do QRSH, and QRSH will get you an interactive session. I'll go over that a little bit later. So QRSH, you have to let that run for a few minutes. And then you'll get back ultimately a command prompt. And you'll notice the only thing that's changed is it's still your username, but it's now at some computer starting with N, and that's a compute node. There's 2 ways to run jobs on the Hoffman cluster. You can run interactively, or you can run from a script. If you're going to run something that's going to take a long time, I highly recommend learning how to run it in a script, because you can put your script in the job queue on the cluster, which means you don't have to sit there and babysit it. So just as an example, last night I was running a small job on the cluster, but my internet went out for a minute or two, and so I got disconnected from the cluster. My job actually got killed. So you'll lose your connection, you'll lose your node if you get disconnected. On the other hand, if it's running in the job queue, then basically the cluster is just going to wait to have the resources available to run it, and then once it has the resources, it'll run the job, and you'll just get an email notifying you when the job starts and when the job ends. So this one is completely insensitive to you being connected or not. With QRSH, you can set a few options. I'll show everybody how to do this a little bit later because we're actually going to QRSH and we're going to set a few options. All right, and this is probably this is probably one of the more important parts to understand. So this is a review of the Linux file system and understanding how to navigate it. And this kind of seems to be a lot of people's biggest hurdle when they get started working in a command line environment. So the Linux file system is just a big tree structure, and you can navigate it as a big tree structure. I have a lot of stuff missing from here. So obviously you can have more than 2 branches at each point, like you see here. And in fact, just from home, there's basically there's going to be a folder for every starting character on an account. So there may even be numbered folders here. So we have A through L, we have N through Z, and in the middle is M, which I specify because that's where my account lives. So on the Hoffman cluster, pretty much everything you're going to be interested in is going to be in the /U. So slash is pronounced root in a lot of cases, or if you're going specifically to this part, which is the kind of the highest or lowest level depending on where you're counting folder, but it's the origin of everything. It's called people will call this root. And so under root, you have /u on the Hoffman cluster, which is where the basically user stuff lives, or where stuff for users is. Under there, you'll find a few things, including home and scratch. So in home, you'll see there's a bunch of different letters, and that's the first character of login name. So under /m, you'll see /m Weinstein, and that's, or M Weinstein in this case, and that's my account. And in there, for example, you'll find program.py and data.txt, which is just a program file and data file. So let's start off talking about how to navigate this thing. So whenever you log into the cluster, unless you have something else set up unusually, you will log in immediately to your home folder. So you are here. If you type in pwd, you'll get back the full path. If you type in ls, you'll see the contents of where you are. If you type in ls space ... you'll actually get the contents of this folder. So you're saying list the contents of the folder directly above. In this case, it would be M Weinstein. In reality, I get every folder that starts with M, every folder that's under this one. If I type in, so I'm still here, if I type in program.py with data.txt, this is actually going to return an error because it's going to say the executable is not found. So it turns out if you're trying to run something, there's basically two ways to have it runnable. If you have it on your path, that's something you can do. I'm not going to get into that because that's kind of outside the scope of this class. The other thing you can do is you can do this, which is just /program.py and then with data.txt. And it'll do exactly what you want. It'll run program.py with data.txt as the input. So if I do cd dot, this is actually a useless command. And that's actually saying change directories into my current directory, so that fundamentally not changing directories, so it'll do nothing. If I do cd dot dot, that's actually going to move me. So now I'm in the folder containing all the home folders starting with M. And if I do cd M Weinstein, that'll take me back into my folder. And this is what's called a relative path. So when I give this value here, because it doesn't start with a slash, I'm saying change directories into some folder relative to this one called M Weinstein. And it's going to know, for example, that that's one folder below. Now in this case, I'm doing cd and then /u/scratch/M Weinstein. So the easy way to understand this is when you're dealing with a relative path, it's looking at where you are right now and moving relative to that. So if I do cd into M Weinstein from here, it'll go this way. If I do cd into M Weinstein, but I'm actually in the home folder, it'll look through here and it'll say there is nothing called that, you're making a mistake. So anyway, if I do that, it'll take me back to my home folder. Now for an absolute path, which is one that starts with a slash, you're going to interpret it a little bit differently. So this is starting with root, and it's going into /u, and then scratch, and then M, and then M Weinstein. So this will take me directly to my scratch folder. And again, this is called an absolute path. If I do cd and then the tilde, so this is a shorthand for my home folder, but this is functionally an absolute path. That'll take me back home. And if I do cd and then $scratch, so $scratch is a command line is an environmental variable that exists in on the Hoffman cluster. That'll take me directly to my scratch folder, and that's how we navigate the that's how we navigate a Linux file system. All right, so quick question: who out there already has a program like FileZilla or Cyberduck or something along those lines installed on their computer? Okay, I see a few people. I guess a more important question, so people put your hands up, you can put your hands back down. All right. So I guess the more important question, who out there does not have some kind of a program for transferring data to and from the Hoffman cluster? Okay, so I have a couple people who do not. All right, let me get a window open here. All right, people should be seeing an internet browser window now. So you're going to find Cyberduck. You can do a quick Google search for it. It'll come up pretty quickly. So go to Cyberduck and then go to download. And you'll get to this page. Be careful. So Cyberduck page has an ad up here. They've done a little bit of a better job of it, but occasionally this ad will actually have buttons saying download on it, which makes you think you're downloading Cyberduck. And you're totally not. It's usually a computer virus. So in terms of downloading, if you're on a Windows machine, you're going to want this. If you're on a Mac, you're going to want this. If you're on a Linux machine, you probably already know how to transfer files over SSH. And for Linux machines, I think the standard is FileZilla. But if you have Windows, you'll want to download the Windows version. If you have a Mac, you'll want to download the Mac version. And I will give everybody who needs it a minute or two to get that downloaded and installed on your computer. All right, so I'll give those people a minute or two to do it. So say I'll pick this back up around 10:40. So I'll give you about 5 minutes to go through the install process. So don't just download it, actually get it installed on your computer and boot it up. And once you have it booted up, I will walk you through the setup process. All right, so quick show of hands of the people who are installing Cyberduck. Is there anybody who has not yet gotten Cyberduck up and running? All right, I don't see any hands going up, so I think we're probably good to continue. All right, are people seeing my slides again? Oh, and I have a question. Do we need to get the, do we need a registration key for Cyberduck? So Cyberduck is distributed as free software, so they don't charge you to use it. You will get a nag screen when you finish that basically asks you to pay for it. That being said, the guy writes Cyberduck, he doesn't charge for it. So if you have the money and you feel so inclined, you're getting a lot of use out of Cyberduck, by all means, you know, get a paid version and kind of make it worth their while to keep maintaining it, because it's definitely a useful program. I know I've definitely done that before. All right, so when you get Cyberduck booted up, you'll see a screen that looks kind of like this, except you won't be seeing existing bookmarks because you have a new install. So you're going to go up to bookmark, and on Mac this is going to be a little bit different because Mac's UI is a little bit different, but you're going to go to bookmark and then new bookmark. So you'll click on that. Once you hit new bookmark, you'll see a bookmark appear behind it, but you'll also see something that looks like this appear. So it'll just say FTP and it'll have basically no information in it. So the first place you're going to click is up here at the top. And that'll have you with, that'll give you a pull down menu. You're going to select SFTP, which is SSH File Transfer Protocol. So you'll click on that and you'll see this window changes slightly. So we're just going to go through this and fill out the fields we need to. So you're going to give it a nickname, and the nickname we're going to give it is Hoffman2 home folder. Your server is going to be this, and I wrote it out here in bigger letters because I know a lot of people have trouble seeing it over here. So it's dtn2.hoffman2.idre.ucla.edu. DTN2 is just saying specifically connect to the Hoffman cluster through one of their data transfer nodes. So these are basically higher speed nodes for data transfer, and they're not for computing. They have much better network connections though. So the username, this is my username. You're not going to want to use my username because you won't be able to log in. The port should be 22 already. So you probably won't need to change this unless it's something other than 22, but it should default to 22. So again, username, you'll put in your username. Password, this is going to be your SSH password that you use to log into the cluster. Oh, I got a typo there, I'll fix that later. So yeah, this will be your typical SSH password that you just use when you log into the cluster. And then the last thing we want to change, and you may not see this, so if you don't see this, you'll just click here on more options, and that should make this menu appear. You're going to set the path to /u/home/ the first letter of your username / your username, and that'll be your home folder. Once you've done that, you'll go up and you'll just click on the X, so that'll just close it, and it'll automatically save the new bookmark. So you'll have this new bookmark called Hoffman Home Folder. You're going to right-click on that, and that'll get you a little menu that gives you a few options. So you can either connect, you can make a new bookmark, which is not exactly what we're going to do right now. You can delete the bookmark, which we definitely don't want to do. We can edit the bookmark, which is useful if you made a mistake. But the thing we're going to do right now is we're going to duplicate the bookmark. So when you duplicate the bookmark, it'll create a duplicate of it pretty much immediately, as you see here. And you'll have this window appear again. And we're just going to do 2 very quick changes to this in this window. So this is an exact copy of the previous bookmark. In the nickname, we're going to change home to scratch, because this is a connection directly into our scratch folder. And then in the path, we're going to change the home to scratch again. So those are literally the only changes we have to do here. And this will give us the ability to connect to Hoffman directly into our scratch folder rather than have to navigate it when we connect. Navigating to the Scratch folder in Cyberduck is annoying. Okay, the other thing I'll talk about a little bit is dependency hell. So are people out there familiar with QIIME, or have you guys heard of QIIME before? QIIME? Okay, good. Okay, a couple people say no, a lot of people say yes. QIIME is kind of the it's kind of the basic standard for microbiome analysis. QIIME itself is actually not so much a program as a framework for connecting different microbiome applications. So QIIME has a bunch of programs built into it, and it actually has some really great plotting features for microbiome data. So the course previously was QIIME-centric. I'm actually working right now to get us away from that, because it turns out on the Hoffman cluster, you can only run QIIME. The latest version of QIIME you can run is 1.9. QIIME is on version 2 point something now, and they've basically said that QIIME 1 is deprecated. So until the Hoffman cluster makes it possible to run something else, I basically want to go. I'm modifying slides right now to teach you a different program called Data2, because that is current technology, and it's actually even a different way of calling microbes. So this is just a really cute XKCD comic that kind of describes a scenario that developed where there's just so much weird stuff that the guy's done while he's building on his computer that it's a gigantic mess and a bunch of interrelated dependencies. This is actually a big issue with QIIME. Because QIIME runs a bunch of different programs, QIIME has a bunch of different dependencies, which means installing QIIME is really, really terrible. So there's a couple ways we can deal with this. Compartmentalizing is probably my favorite way. So virtual machines, these are actually running a virtual machine on a computer, basically saying, take 2 CPUs and a certain amount of memory and allocate it and make a virtual computer out of it that's running completely isolated. These are nice because they are really, really isolated. They're kind of annoying because as long as they're running, they take all their resources. And they're very heavy for storage, and they basically occupy as much possible resources as they can, or as they're allowed to. There's another approach called containers. And containers are, I think, probably my favorite solution to this. In fact, you'll see with a lot of programs I write, I actually write them I write the program and I write a container to run the program. Containers are great. They can be distributed quickly. There's Docker and Singularity. So Docker is kind of the standard. And I think a lot of people have heard of Docker, at least talked about by tech people, because tech people seem to like it. I think as good as Docker is for tech, it's even better for science, because you can now not just replicate somebody's computer program, you can replicate their entire environment and make it easily portable. Singularity is just a more secure version of Docker that is designed for these shared computing environments like the Hoffman cluster. And I think Hoffman will soon be supporting this, if they're not already. So containers are also pretty well isolated. They're not as isolated as virtual machines. There are potential issues with some containers of somebody like accessing memory they shouldn't. They're much later for resources. So containers only going to take the resources it wants to take, or it needs to take it for what it's doing. And then there's environment managers. So things like Python's virtualenv and Anaconda. And on the Hoffman cluster, you'll see module load showing up. These are super light. Module is available on Hoffman too. These things are poorly isolated because they're basically just changing your environment around to pull in some other things that they weren't before and to sometimes remove things. So sometimes the environments don't really isolate, and you can have stuff in your environment you don't realize or want. And occasionally it will still run into conflicts with your existing environment. So these will work, but they have to be configured very carefully. All right, now I'm going to get into the course goals for just a moment. So what are the goals after this class? You should understand the basics of sequencing and operating in a Linux environment. you should, after this class, be able to understand the different methods of organism identification. You should be able to understand the differences between targeted and shotgun sequencing, as well as their limitations. Understand and execute organism identification using a database. So you should be able to also understand what the problems are with the databases, because our databases are definitely not perfect, and there's actually a lot of bias built in. And you should be able to understand and execute comparison of different microbiome samples. So the point I think I'm probably going to end up taking people up to in this course is being able to identify microbes and assigning taxonomy to the sequences. So figuring out what the sequence likely came from. All right, and now's where we get into the fun stuff. So we're past the point of review, and we're going to get into types of analysis. And actually, before we get into that, because I think that's going to be the last thing I cover today, it's 10:49. Let's resume at 11 o'clock. This is a really good place for a stretching and bio break. And yes, you'll find I'm actually very strict with the with calling for breaks about every hour or so. So I obviously can't tell if anybody out there is pregnant right now, but I've actually had quite a few students taking my class while they were pregnant, and I learned that calling for restroom breaks every hour is a mercy for some people. So, anyway, 10:50. Let's pick back again at 11 o'clock, and we'll go over types of analysis. And this is I think what's probably the most important section of this class, because a lot of people will run analysis, and a lot of times when I ask people why they're running a kind of analysis, especially when it makes no sense, or when I look at what they're doing, like, it doesn't make sense why you're doing this particular analysis on this type of sample, they say, well, somebody I know was doing this exact analysis, but it turns out they were asking a completely different question. So understanding your analysis and understanding what kind of sample you're looking at is kind of one of the keys. And with that, I will see everybody in about 10 minutes. I think everybody should be back by now. All right, and I got a question during the break. So question is, I installed QIIME on my laptop and it's working, but I have difficulty to log into the Hoffman cluster. Do I need to go to Hoffman too? Should everybody install Cyberduck? So the answer to those questions, if you have QIIME on your laptop already, or if you have some other computer you run this thing on and you have QIIME already installed, especially and you already know how to use it, that's perfectly fine. QIIME is a good set of programs. QIIME is a good system. So definitely feel free to. As I mentioned before, I will, I'm actually leaning towards teaching Data 2 in this class for the simple reason that the only access I can assume people in this course have is to the Hoffman cluster, and I'm trying to avoid teaching QIIME 1.9 because I don't want to teach software that people should generally not be using as their core analysis program when QIIME 2 is kind of considered the current standard. I do know there's R on the Hoffman cluster, and through R I can run something called Data 2. And the truth of the matter is Data 2 is also a newer system for assigning taxonomy that I like a lot better personally, and I'm about to go into why. As for Cyberduck, if you're going to use the Hoffman cluster, you should either install Cyberduck, or if there's another program you like for transferring files to and from servers, you're welcome to use that. So if you have FileZilla for talking to the Hoffman cluster, you're fine with that. And I know for Mac, there's a very large number of file transfer programs. For PC, there's not quite as many. Or likewise, if you have PuTTY installed and you have WinSCP and you're used to WinSCP, that's also perfectly fine. I recommend Cyberduck because it's one that I already have instructions on how to set up. So if you don't have anything already set up, Cyberduck is great. All right, now on to types of analysis, which is, in my opinion, probably the most important unit of this class to understand. This will be basically describing what we're going to do to the data that we'll be getting, and we'll be actually starting to run analysis tomorrow and understanding where the data come from and that sort of thing. All right, so the first question when you're doing any NGS study, and I don't care if it's microbiome, RNA-Seq, epigenetics with like methylation-Seq or ChIP-Seq or whatever, it doesn't really matter. You first have to ask, what do you really want to know? Because if you want to get your desired answer, you really have to ask the right question. Just as an example, I've had labs that were getting my assistants doing RNA-Seq. And then after we do the RNA-Seq, they say, oh, I was doing RNA-Seq in tumors. They say, can you check the tumor for mutations? And my response is usually, sure. Do you have exome data? And they say, no, we want you to check for mutations in the RNA. RNA, I can occasionally find mutations just by looking at RNA-Seq if I'm doing a very limited check, but variant discovery in RNA, despite the fact that it sounds like it should theoretically work, never works that well in real life. So whatever it is you want to know, make sure you're getting the right samples and asking the right questions and doing the right analysis to get the answer to what you want to know. So in the case of microbiomics or metagenomics, it gives us a few options for what we examine and what we look at. And what we look at is going to determine what we see. In the realm of DNA sequencing, we have essentially two options. We have sort of whole genome shotgun sequencing, which is basically just get all the DNA we can possibly get out of the sample, fragment it, and sequence the fragments. So this will give us a few advantages. Shotgun sequencing will pick up whole genomes and it'll pick up all the genes. So we'll know what the microbe is capable of. We'll know, for example, if we have a lot of microbes with genes for fermenting fructose, which can be important in the intestine. One thing that's annoying here is unless you use some fancy tricks during prep, you're probably gonna sequence whatever host DNA is in there as well. So if you're not interested in host DNA, but you're sequencing, for example, human stool, you're gonna pick up a lot of human DNA in there because it passed through human. You have a much larger genome space. What I mean by that is your potential genome is essentially all the microbial genome out there. That makes for much harder informatics, you need more computing power, that kind of thing. You also can occasionally get confused. There's a lot of genomic elements in bacteria that are notoriously mobile. So if you sequence a transposon, you're pretty much going to mask that out because transposons are, well, they're pretty much defined by the fact that they're mobile and they jump around. Alternatively, there's some genetic elements that are mobile, but they're not notoriously mobile. So for example, the virulence factors in Salmonella, they actually came in via bacteriophage, and they're pretty much always associated with Salmonella. But because they came in with bacteriophage, they can occasionally jump around and they can occasionally show up in some other microbes. But depending on the database you used and some of the filters you put on there, if you see that, you're just going to think you have pathogenic Salmonella. Targeted sequencing can get around a lot of these problems, which is awesome. But targeted sequencing also has some disadvantages. So targeted sequencing, you're going to pick a gene that's common to a bunch of your species of interest that should have some common regions that you can prime to, and it should have some conserved regions you can prime to. It also needs some variable regions that you can use to differentiate between the gene of different species. The best you can really do with this is typically inferring function. And I'll go into that in a bit, but basically you're only sequencing one gene or one region. So you're not actually seeing any of the other functional genes. So the best you can do is just infer that, for example, E. coli tends to behave a certain way. So I'm seeing E. coli, so I can infer they're going to behave a certain way. All right, some of the other options. These are some of the more exotic ones that are less in use now, but they're still being explored. And these are non-DNA options. So there's RNA sequencing or metatranscriptomics. Transcripts are awesome because you can actually tie them back to a gene of origin. So you can occasionally also figure out even what species they came from, but you can pretty much always figure out like what enzyme you're looking at or what other gene you're looking at with a transcript. So that's awesome. If a gene is not being expressed or not being expressed well, there's a decent chance you will never see it in a transcriptome because it has to be expressed to show up in a transcriptome. You need to remove ribosomal RNA. So ribosomal RNA makes up a lot of the RNA cell mass, a lot of the RNA mass in a cell. If you have a lot of ribosomal RNA in there, you'll get a lot of reads from ribosomal RNA and not a lot from the messenger RNAs, which are what you want. But what's really cool is you will get quantitative gene expression. So as I mentioned before, kind of the lowest level of information you can get is targeted sequencing, which is really just saying who's here or which microbes are here. And occasionally, if I know names, I can kind of make inferences about what they might be into. So for example, if I see the name Elon Musk, I'm probably dealing with Elon Musk. Like, dude makes Teslas, he's in the news lately for complaining, that sort of thing. If I see the name William Gates, yeah, I might be talking about Bill Gates, the millionaire. But it turns out there's a lot of people named William Gates out there. There's also William Gates, the basketball player. There's like William Gates, the mechanic. William is a common first name. Gates is a common last name. So I can't really make a big inference about what they're doing. So if I see something uncommon or something with very well-known specific roles, I can make some inferences from the name or from, in this case, targeted gene sequence. If I see something that is, kind of more versatile microbe like E. coli, I can do a bunch of different stuff. I don't necessarily know what they're capable of or what they're doing. If I do whole genome, I know what they're capable of now. So I know which genes they have, and I can kind of make some guesses as to which genes they don't have because I would have expected to see them. With RNA sequencing, though, I don't just know who they are, and I don't just know what they're capable of. I know what genes they're expressing, so I know what they're doing. And that can be really key because sometimes even if they have a gene, you'll figure out that it's turned off. And then finally, there's metabolomics. This is still a very young field. It's exciting. I did a lot of metabolism research, so I'm super excited to find out what happens here. Metabolomics also can kind of be a nightmare. So metabolomics, you're going to be doing your analysis probably on a mass spec. So I talked before about gene space, how with shotgun sequencing, you're looking at basically all the genomes of all the microbes. In this case, in chemical space, there's so much more diversity even. At least if I'm looking at DNA or RNA, I know kind of how the molecules I'm interested in are going to behave, and I know how to work with them, and I have really well-established techniques. In the chemical space, I could be looking at nucleic acids or even just the bases. I could be looking at lipids, carbohydrates, amino acids, proteins, whatever. And the problem, for example, is some carbohydrates tend to oxidize. Lipids, some lipids are super stable. Some lipids are extremely volatile, like if I have butyric acid producing bacteria, that butyric acid is going to start evaporating really quickly from my sample. Some other lipids may be chemically unstable, and they may just oxidize if they're exposed to oxygen for any amount of time. But the cool thing about this is you move even beyond what the microbes are doing, and you can start asking about questions about what are these microbes breaking down or building. All right, so let's focus on DNA because that's obviously the focus of this class. Metatranscriptomics is awesome, but I'm not going to get into it for this course. So DNA provides us a few options. Here we have a bacterial cell. Judging by what appears to be potentially a thick cell wall around it, probably gram-positive, but whatever. The thing we're focused on is we have a cell, and inside the cell we have DNA. So that DNA is going to come in the form of a bacterial chromosome, and it's a very large piece of DNA. And I call this typically, if I'm talking about in terms of just genomic information, I'll call this kind of the core genome. A lot of bacteria also routinely carry around plasmids. These are episomes, so things that are outside the core genome. And these plasmids can sometimes be present and sometimes be absent. If I get rid of the cell, then I'm just left with the DNA, which is what you typically do for analysis. And if you're doing shotgun, or if you're doing, full shotgun metagenomics, you're going to detect both of these things. Now, if you're doing again whole genome shotgun, if you get enough reads, you can actually assemble a genome out of them. So you can basically take all these reads, run them through an assembler, and it'll use these contiguous regions and eventually assemble contigs. And you can assemble the contiguous regions and into a full-on bacterial chromosome and bacterial genome. This is actually one place where long read technology really helps. So since you're trying to assemble contiguous regions, assembling a bunch of really long reads together is much easier than assembling a bunch of really short reads. And as I mentioned before, some of the long reading technology, especially things like nanopore, tend to have high error rates. So what you would wind up with this is you'd have a very long contiguous genome that you can build up fairly quickly from nanopore data, but it would be full of errors. So what a lot of people like to do is they'll do both short and long reads. They'll do long reads to assemble basically a scaffolding or kind of a backbone of the genome that they know is rich in errors. And then they'll start aligning short reads to it, and they'll use the short reads for what's called polishing, which is where they come out with a first draft of the genome that, again, is full of errors, but is at least a contig. And they'll start using the short reads to basically make a second draft of the genome immediately, where they fixed some of these base call errors. All right, so this is where we get into kind of a little issue with how bacteria are identified and how they're named. And the simple fact is that some species were differentiated based on their plasmids. So this is Bacillus cereus. Bacillus cereus is kind of a minor pathogen. It's found throughout the environment. It sporulates, so it hangs around a lot of things and it can stay pretty permanent. The main place where I've heard of people contracting this is basically this will make some people sick if the spores are on, I think fried rice is a common source of it. So if the spores germinate on fried rice that was improperly stored or improperly processed, it causes basically food poisoning, diarrhea, dysentery type stuff. It, unpleasant, but it kills almost nobody. It's just unpleasant. So it turns out that that this core genome, if I keep it entirely the same, and I swap out a couple of the plasmids, so now there's two specific plasmids in here. I think it's PX01, PX02. It becomes Bacillus anthracis. Anybody out there familiar with this microbe? So quick show of hands, who recognizes this name? Yeah, B. anthracis. So I'm seeing a lot of hands come up. I'm guessing way more people recognize the name B. anthracis versus B. cereus. And it turns out that when people actually did the sort of sequencing of the bacterial chromosome or the core genome, it was really quickly figured out that actually B. cereus and B. anthracis, the line between them is not very sharp. And in fact, some B. anthracis strains came back as this should actually be classified as a B. cereus based on similarity. And that's because when microbes were first being studied, they were first, initially some guy named Leeuwenhoek in the Netherlands built good microscopes, and he could actually see microbes, and he said, oh, it turns out a drop of rain has, a little drop of water, a drop of rain, or whatever else. It's full of these things that are actually kind of alive. And they were a curiosity for a little while. And it wasn't until Pasteur and Koch came along where they said, oh, these things aren't just alive, they're actually causing disease. And then for a while, microbes were just associated as like these things that were causing disease. And that's still very much coloring our understanding of microbiome now, even though thanks to microbiome research, it's changing where everybody thought that the relationship between human and microbe was antagonistic, or at the very least somewhat kind of exploitative, where microbes were basically breaking down our bodies after we died, and we were harnessing microbes to do things like make our beer, raise our bread, all that other good stuff. So early on in microbiology, things were just being named by stuff that people could detect, and things that people could detect included things like what was the shape of the microbe, what did it do when you cultured it, did it make people sick, and they didn't really understand the microbial genomics at the time. So that led to situations like this where we have B. anthracis and B. cereus, where the core genome is actually pretty much the same. B. anthracis, the thing that really defines B. anthracis, is actually the two plasmids that make the toxins that seem to have a critical role in its pathogenicity. And if it loses one of those two plasmids, in fact, if it loses one for something called protective antigen, it actually becomes B. anthracis vaccine strain. So the response to anthrax is a bit different than the response to B. cereus. Also, totally not adequate PPE there if there's B. anthracis in that tube. But what can you do? You can't really get an N95 mask right now. So DNA also provides a few other options. So what if you had a scenario where maybe instead of doing the whole genome, which can be kind of tricky and is very intensive for data processing and that kind of thing, you can instead just focus on a small part of the genome that you could use kind of as a barcode or identifier for the microbes, almost like a name tag. And it turns out there's some good regions for that. So what you really want is a region that kind of has this pattern where if you go along the length of the gene, you'll see that you have some areas that are very highly conserved. You'll also see that you have you'll see interspersed in their regions that are also highly variable. And this gives us a structure that we can work with. And the reason we can work with it is these highly conserved regions are useful for priming. These highly variable regions are useful for identification. So for example, I can prime in the conserved region, I can read across the variable region. And if I know what microbes tend to have this exact variable region sequence, then I basically have a tag for them. I can't necessarily say anything about them beyond just they have the variable region of this microbe and what microbe they probably are, but I can at least start making some inferences about who's there. And if I know who's there, I can at least try and infer what they're doing, even if I can't say exactly. And as an added bonus, if this region in between these two variable regions is small, I can actually prime all the way over here, and I've actually tied together two variable regions right now. So anybody out there familiar with 16S sequencing already? Okay, so for the people who are familiar with 16S sequencing, you probably realize that what I'm describing here is 16S sequencing, and the genomic architecture I'm describing is the architecture of the 16S gene. And you'd be right, but that's actually not exhaustive. For example, certain common heat shock proteins also have patterns of conserved and variable regions. And I think Wayne's probably the only one who's looking up right now because I know that, for example, in water microbiology and when analyzing pathogens, there's certain heat shock proteins that people like better because those heat shock proteins actually give you better resolution of specific pathogens that show up. And again, this is really popular in water and a few other places. So 16S is by far the most popular gene for doing this, but 16S is not the only gene, and there's certain other genes that that are very good for this as well, especially if you're very interested in a specific group. So I think, for example, the heat shock protein can give you much better resolution within the world of Staphylococcus. So what do you look for in a targeted sequencing gene? Number one, it has to be sequenceable. So if the gene has some crazy GC content, you don't want to use it. You need a pattern where you have highly conserved regions surrounded by highly variable, or you need highly conserved regions surrounding highly variable regions. It has to be present in your target species, obviously. And if you want to focus in, you want it ideally absent in non-target species. So for example, 16S is also good because, and heat shock protein is good because you get those genes in microbes, and especially microbes you're interested in. But 16S sequence doesn't really exist in archaea or eukaryotes. So that'll kind of limit you to bacteria. Depending on the primers you use, you will get a little bit of amplification of 18S sequence from eukaryotes, but it'll be kind of much more controlled in that case. The other good thing is because a lot of these target genes are used by a lot of people. For example, there's a huge database of 16S sequences out there. The problem with the 16S databases is not even that we don't have necessarily enough, it's that we actually sometimes have too much and people have deposited garbage. So, and again, you want a large existing database if possible. So what are we actually sequencing for 16S? We're sequencing this part of the ribosome. So it turns out ribosomes are mostly made of RNA, and we're just sequencing the smaller portion. All right, so I'm going to go through 16S analysis, and then after that, I believe I'm going to call it a day, and we'll pick up again tomorrow. So 16S sequencing challenges. We know that sequencing is imperfect. We know that Illumina makes about 2% base call errors. Nanopore makes even more, but it does really long reads. So Nanopore has the advantage you can read an entire 16S gene end to end, whereas Illumina, you can only read some regions of it. And the errors aren't always evenly distributed. That's why you can actually build an error model and you can say A to T changes are more common than T to A changes by mistake, that sort of thing. So the real problem with errors is if you make a single base error in a 16S sequence, because 16S you're calling based on just a few nucleotide changes, you don't want to confuse a read error with a new species. That would obviously be bad. But we know there's going to be errors, and we know that if we don't deal with the fact there's errors, those errors are going to look a lot like new species or increased diversity. So there's two possible ways of essentially dealing with these kind of errors. One possible way is you can kind of blur a little bit, where you're basically saying, I'm not going to worry as much about single nucleotide differences. The other way is sharpen. And for anybody who's seen Blade Runner, you're probably, the phrase going through your head right now is probably enhance as you look at this. So clustering or blurring, you're basically going to cluster together microbes that appear to be very similar. So here you see I have clusters of microbes, and here we have, these guys are shaped like bifidobacteria. So here we have kind of the light bluish bifidobacteria. Here we have the brightly colored bifidobacteria. I think these are Diplococcus or whatever. Here we have just all the Diplococcus that seem to be pretty darn similar. Here we have dark colored bacilli. Here we have light colored bacilli. And here we have the flagellates, which apparently were all similar enough that we want to consider them a single group. And this basically forms clusters. And the great thing is, even if a couple of these were actually present just due to being sequencing errors, if we treat them all as a cluster instead of a single microbe, then a sequencing error is a couple of sequencing errors, like let's say these two are super uncommon and they're actually caused by sequencing errors based from this one. It doesn't matter because they're just a cluster. So the sequencing errors get lost in the cluster because we're essentially blurring our image a little bit. Occasionally clustering has a little bit of a disadvantage. So if I'm building my clusters de novo, rather than using a reference for building my clusters, let's say these guys come in, and these guys are dark colored, but they look kind of similar also to some of these guys here, like especially this one. That can actually change your clustering if you're clustering de novo. So the addition or removal of microbes can actually change your clustering pattern. A cluster is called an operational taxonomic unit, or an OTU. And again, it's just a cluster of microbes that you form. OTU picking can either be guided by a reference, where basically some of these microbes in here would actually be reference sequences. And you would be using those reference sequences and building your clusters around what cluster is close to the reference. I'll get into more detail on it tomorrow. But basically there's what's called de novo or reference free. There's open reference, there's closed reference. Closed reference, everything is limited to just the reference. Open reference, you're going to try and cluster things to a reference species. And if you don't have a close enough matching reference species, you'll then de novo cluster it. And de novo clustering or reference-free clustering is just you build the clusters out of the sample. Obviously, going reference-free is very computationally intensive, and it's not deterministic. The clusters you form depend on what's in the sample, and if something new is present or something that was there before is missing, your clustering will change. All right, so how does our OTU approach work? We're going to combine extremely similar species. So for example, 97% identity will typically be called for species similarity, although a lot of times this will still just get you down to genus level. So to do this, the workflow is pretty simple. You do any necessary pre-processing. You're going to pick your OTUs. So that's that clustering process I mentioned before. You're going to extract representative sequences from each OTU. So each of those clusters, you'll kind of come up with a consensus sequence that describes the OTU. And you can use that to either describe it, or you can use it to compare it to a reference and figure out what it looks most like. And then you're going to do some kind of further analysis, whatever you happen to want to do downstream, whether it's diversity or whatever. So there's another approach. So here's where I'm going to ask you a question. But what if we considered frequency? So going into this Diplococcus group, let's say we saw this thing, 10,000 counts. So we got 10,000 sequences that were exact match to this. We got 15 counts that were a match to this, and we got 2 counts that were matched to this. Oh, well, that's interesting because these two are pretty rare. And we know there's some error frequency. So what if we considered frequency of the reads that we detected? What if we also considered the differences between them? So for example, we see this one 15 times with 1 base difference. We see this one 2 times with a 2 base difference. And what if we consider the error model for the run? So based on error modeling, we see 15 counts of this 1 base variant. We expected to see it by our error model about 18 times, so about what we expected. Who thinks that now we can say this thing probably doesn't exist and is probably just artifact? Okay, so I see people going to raise their hands, and that's good because, yeah. Basically, in science, if you want to say that something exists or it's real, you have to provide support for it. The null hypothesis on this would be this doesn't exist. This is actually just sequencing error. In this case, it looks like it's a pretty predictable sequencing error. Likewise with this one, we saw 2 counts of it. We expected to see about 1. This is also easily explainable within sequencing error. So what if we start taking this into account? We can start saying things like, this is bogus, this is bogus. These are just artifacts of our sequencing. Neither of these things that look like they were detected sequences really exist. So now we've actually turned that 3-organism OTU, where we just have a representative sequence, into a very specific 1 organism that we can say exactly what its sequence is. And it's real. And the nice thing is, not only can we say it's real, we can say statistically, this was almost impossible to be just sequencing artifact. This is a real sequence from our sample. And you're going to say, this looks a lot functionally like our operating taxonomic units, our OTU approach. So our OTU approach just said, these ones look really similar. Let's just lump them into 1 in case they really are just, 1 thing with some artifact. And that's valid, except that we can also sometimes get some serious advantages. So number 1, previously I said we had 1 sequence for that organism, and we could be confident that was a sequence that really existed. What happens if I start changing these numbers? So here we have 150 counts, we expected about 18. Here we have 2,000 counts, we expect about 1 by mistake. Now all of a sudden we can say, statistically speaking, these are probably real. So that 3 OTU cluster is now not just three organisms represented by 1 consensus sequence. It's 3 organisms represented by real sequences that represent those microbes, and that we can statistically support as existing in our sample. So this is the amplicon sequence variant approach, or ASV approach. This is primarily done using a program called Data2, although there's some other programs out there like Deblur that can do it as well. But this is kind of the latest and greatest for identifying which microbes are really in a sample from target gene sequencing. The workflow for this is a little bit more complicated because, as you probably saw, it's not just trying to blur out the photo a little bit. It's actually trying to actually enhance the image. So just like before, you do your pre-processing. You're going to do trimming and filtering, which a lot of people consider part of pre-processing. In this case, you have to build an error model for the forward and reverse read. So in order to predict what the likelihood is of seeing any given change in a certain quantity, you have to figure out what those probabilities are, and doing that requires building an error model. You're then going to merge your forward and reverse reads into a single amplicon at some overlap region in the middle. So this is where having those really long MiSeq reads gets helpful. You're going to count the occurrences of each exact amplicon. So now you're handling each sequence as just an exact match or not. Then you're going to use an error model, and you're going to start asking questions like, which of the rare amplicons can be explained as a more common amplicon with a sequencing error? And if it looks like it's an unsupportable amplicon, you're going to end up eliminating it. So if you say statistically, we expect to see this amplicon just by our error model at a pretty high rate, you're going to throw it out. And now you can go forward and you're doing your analysis. Instead of using representative consensus sequences from what may be 1 microbe or the errors, or maybe a group of microbes that actually existed that you just couldn't differentiate, you're going to go forward and do your analysis saying, I have the sequence, I know the sequence is real, and it represents a microbe that was in my sample at a certain relative abundance. All right, and I think that's where I'm going to cut off for today. So anybody who can't access the Hoffman cluster, please try and get access to the Hoffman cluster. Just out of curiosity, how many people in this course have already run a microbiome analysis doing 16S? Or if you haven't run it yourself, at least participated and kind of had exposure to it. I know people in the Shao lab, for example, probably at least see this in the lab meeting. Okay, good. A lot of people have had exposure to 16S sequencing. All right, so for people who've had exposure to 16S sequencing, how many of them was it exposure to ASV? Okay, it looks like I see one person who still has a hand up for having experienced ASV. How many people's 16S sequencing exposure was to the OTU approach? Okay, that's kind of what I thought. So a lot of people in this class, I think, are already somewhat experienced in QIIME, have already seen it. So that kind of tells me where I want to go with this. So I'm going to put together our actual hands-on exercise. We're going to actually use ASV approach on some sequences. So we're going to see how to run Data2. And you can run Data2 through QIIME2. It's been integrated in there. I'm going to show how to run Data2 just through R, so kind of running, kind of what I call running Data2, just sort of basically naked Data2, which is running Data2 as an R package. Has anybody out there run Data2 before? Okay, excellent. So that's something I can show people that'll be new for them. Okay, cool. So I'm in the process of making that right now, and now that I know that everybody, that would probably be more interest because it's kind of a new approach. Just a quick show of hands. Who came into this class not knowing the difference between, well, I guess first question, who came into this class not knowing that the ASV approach existed? Okay, so 8 people weren't familiar with it. Who came into this class having never worked with the ASV approach before? Okay, so pretty much everybody. Now that I've kind of showed you the difference between the ASV approach and the OTU approach, who feels like they kind of understand the key differences between them and the advantages and disadvantages of both? Awesome. Okay, that's been a big thing for me. Is a lot of people are working still with a 16S or targeted gene approach, and some of them don't even know about the ASV approach, and some people know about it, don't really understand what the difference is, and they think that ASVs are just more precise OTUs. Okay, and the last question. Now that everybody here kind of knows about the ASV approach, who thinks they might want to try it one day and see if they get better resolution than OTUs? Okay, perfect. So yeah, so tomorrow I'm going to have us do a basic ASV calling. So yeah, so for tomorrow and Thursday, we're going to actually go through the ASV calling process, and I'll talk about some other issues related to microbiome. One last question before I call it a day. Who out there has actually given a lot of thought to their sample handling and sample prep? Okay, good, quite a few people. So that's going to be an element that I talk about in this class that didn't exist in the previous iteration. And part of that, again, is because I work at Zymo and I deal a lot with sample prep, but it turns out your sample prep methods can completely alter what you see when you do your analysis. So if you haven't seen it before, I'm going to explain to you why probably a lot of microbiome data you're seeing has massive overrepresentation of gram negatives. Okay, so before I go, I have one question from Eva. And that question is, could you provide examples of analysis that would lead us to want to use ASV versus OTU? Like, is one better for microbiome versus microbes in the environment? So that is an excellent question. My general view on this is because ASV is getting you down to a specific sequence that you can statistically support as existing in your sample, I think ASV is pretty much appropriate anywhere. OTUs are good for both the fact that they've been used historically and they're the original method, so if you have to compare, that's definitely something you want to consider. OTUs, especially if you're generating them, so OTUs become a huge pain in the butt if you're dealing with environmental samples. And that's because, you know, if I'm taking a human stool sample and I'm doing a characterization on the microbiome in there, I can build my OTUs very computationally efficiently, you know, with like minimal computing resources by building them around the reference genomes or around the 16S reference databases. And the truth of the matter is, and I'll get to this also a little bit tomorrow, our databases weren't just built to be not biased. If you look at what's in the databases, if I have some E. coli and I throw it and I test it against the database, what I'm going to find is that I'm going to get like the 100 closest strains because E. coli is in the database massively overrepresented because that is where molecular biology started studying life itself and the molecular functioning behind it. If I'm dealing with something that came from a human, especially a stool sample, that's going to be really well represented in the databases because basically if somebody's doing microbiology and they're studying humans, they tend to get more funding. The next place where people tend to get more funding, if I'm studying some strain that's showing up in food, either causing food spoilage or causing commercially important fermentation, like, you know, it's what Boudin Bakery is using to make their sourdough bread, that's going to be in there. And that's going to be really, really well represented because commercial microbiology is also a big money maker. Things that don't end up getting a lot of funding, especially until recently, if I'm doing my sequencing on a cave in the Marianas Trench with a thermal vent in it because I want to see what kind of microbes live in a cave with a thermal vent. The databases are probably going to confuse me more than they help me. And that's because, again, the databases are built around, you know, they were doing stuff with humans, and the bacteria I find on a human are going to be way different than the ones I find in that cave. So any similarity is probably going to be confusing, and it's probably just going to be by random chance. So that's where ASVs are great. ASVs don't use any reference until you're just comparing the ASVs to a reference. Whereas if I try and build my OTU table from that underwater cave using the databases, it's going to be building cave OTUs based on reference sequence that is massively overrepresented for E. coli, stuff that lives on or around humans, and stuff that does food. The other option I have is I can also just build my OTUs de novo, which is perfect, which is typically the approach you would do if you're dealing with an environmental microbiome sample, because you know that the database doesn't really apply to you. So I can do that. The problem I would run into a couple problems there. Number one, de novo OTU building is computationally very expensive. It's slow. It's something that we can't even speed up with bigger computers. It's just slow. It can't be run in parallel, so running it on an 8-core system is going to basically have 1 core very busy and 7 cores very tired, or 7 cores very very unused. So yeah, it's inherently very slow. I was talking before about that issue where depending on what's present in the sample, your OTUs can change because it's clustering de novo. That also means that if I get a dozen samples for my study this week and I do an OTU clustering on it, I have to basically repeat the OTU clustering the next week when I get my next dozen samples, essentially from scratch. So any clustering I do, if I get more samples, I have to rerun all the effort for that clustering. So for Eva, does that kind of help you understand? Yeah. And then compare that, for example, to using ASV, where if I just have ASVs, I'm saying this is an exact sequence of a microbe that I can support as existing in my sample, and this is not some potential sequencing error. So that ASV basically stands forever. All right. And so everyone in this class, do you have my email? All right, I'm putting my email address in the chat. Please feel free to give me feedback on this course. Like I said, it's still developing. So you guys are not just the first students from the revamped course. You guys actually have the opportunity to kind of drive it a little bit for the upcoming students. So please feel free to email me with feedback on it. Obviously, if you just email me back saying it sucks, I'm probably going to throw it in the recycle bin. But if you actually have feedback on ways where you would want to see the course change, or things that, something, for example, that you think is a really important topic that I didn't cover today that you want to make sure gets covered during the course, I can even try and make sure I work that in. One thing, the only thing to keep in mind is if it felt like I was doing too much review on basic material, and you're somebody more experienced in microbiome, then probably the review wasn't for you, it was for people who are less experienced. So that's why I had to go over basic Linux and that kind of thing. Anyway, I look forward to seeing your emails with feedback. I know I ran a little bit longer, which is actually useful for me to know because that's going to help me set the timing for this course in the future. So I will see everybody tomorrow morning at 9 a.m. sharp.
Can I ask you a quick question, kind of about building a reference genome de novo?
Sure.
Yeah, so essentially the gist that I got from what you're saying is that it's very hard and very expensive. So I do need to do that. And I just wanted to know if you had any advice to build a reference genome of my own. If you have any like tutorials that you might know of that would be good to kind of help with that.
So when you say building a reference genome, are you talking about building like a reference genome for one species, or are you talking about building? Oh,
for
okay.
one species, for B theta.
For what?
For Bacteroides beta micron, I think is what it is.
Okay.
Beta micron, yeah. Beta,
Okay, yeah. By the way, I'm trying to remember, which lab are you in again? I know you said at the beginning.
Jeff Miller.
Jeff Miller. Oh, that's right. Yeah, that's right, because you're working on the stuff that Sergey was working on before.
Yeah, the DGR stuff.
Yeah. Okay, so you're trying to... Okay, so I'm not super versed in genome assemblies. That being said, the best approach I've seen to doing a de novo genome assembly is look up how to do hybrid assembly. So what you want to do is you want to take your microbe, and you're going to want to sequence it probably using a nanopore.
Mm-hmm.
And that's the method that I said it generates. Well, the ideal way is, in fact, actually the ideal way, especially for you if your lab doesn't do a lot of genome assembly, there's commercial entities that basically you can just send them your sample and they'll give you back an assembled genome on it.
Okay.
So I know there's certain PIs who are always very much like, I want my grad students to do all their own library preps and all this other stuff.
Mm-hmm.
I completely get that. I want my grad students doing all their own assemblies and that kind of thing. I completely get that attitude. I also know a lot of PIs who realize that if somebody's going to be doing an assembly once, they're not going to be mastering it. So don't bother trying to have them master the technique if they're going to be doing it once. It's actually just more efficient to pay somebody to do it for you.
Okay.
So
gotcha.
And I know Jeff tends to be much more reasonable in that area where it's like he can either have a couple months lead time and have you assemble a genome where you're potentially going to make mistakes or have it be suboptimal, or you can send it off to somebody, send the microbes to somebody with like a check for $2,000 and have them send you back a very nice polished genome.
Awesome. Okay.
And
cool.
because my favorite assembly method is actually with the PacBio, giving you long reads with low error rate. That's sort of the optimal one, but a lot of places don't like to have PacBio unless they're doing a lot of de novo genome assembly. So the other way I've seen, and this is probably what you would want to do if you're doing it yourself, nanopore is easy to get a hold of. So you would do a nanopore to build basically a backbone or a scaffold for the genome that is going to have a high error rate. And you do a hybrid assembly, which is taking that backbone with a lot of errors, doing Illumina reads with lower error rates to basically polish up the errors. And then that would give you also a pretty good genome. And it's a lot less computationally intensive than, it's less computationally intensive than building a genome out of a ton of short reads. And it's more accurate than building the genome just out of long reads with high error rates.
Okay. Gotcha. Thank you so much. I appreciate it.
No problem. If you need any advice on that further, I'm more than happy to help.
Perfect. Thank you so much.
All right. I'll talk to you later.